Hello and welcome to our video lecture on section 3.4, where we will be discussing proteins. And these are the most abundant organic molecules within our bodies and will have the most diverse range of functions of all of our macromolecules. They can transport, have storage, or can be found in our membranes. They'll also serve as enzymes and can even be toxins. And we know that the shape of proteins are going to be critical for its function. Whenever we have a change in temperature, or let's say extreme changes in pHs or exposure to chemicals, they're going to go through a process called denaturation, in which we are going to lose the structure of the protein. Now, sometimes they're going to be reversible because that primary structure, which we'll talk about what that is conserved. Although other times we are going to lose that and it will become irreversible, which leads to loss of function. So let's first go through our learning objectives. We want to describe the functions proteins perform in the cell and tissues. We have some idea of that. Then discuss the relationship between amino acids and proteins, explain the 4 levels of protein organization, and lastly describe the ways in which the protein shape and function are linked. So we first can elaborate on enzymes. We sometimes also call these catalysts, and our living cells are able to produce them. And it's helpful because they increase the reaction rate and therefore are our organic catalysts and help us with things like digestion, breaking down things like carbs. We talked about amylase in a previous video and how it breaks down amylose so that we can more easily absorb it. Each enzyme that is going to be specific to a substrate. And you could see some of those enzymes listed here, and we would utilize these for different things. So for instance, we have amylase. We've already talked about that. Lipase is going to help break down fats. Pepsin is going to help break down proteins as well as trypsin. And we have these 3 different groups of enzymes. Our catabolic enzymes, which I just described, help to break down their substrate. Anabolic enzymes are going to help build more complex molecules from their substrate. So again, adding those pieces together, making them larger. And then our catalytic enzymes are going to affect the rate of reaction, speeding it up. And our proteins are going to serve to be chemical signaling molecules known as our hormones. And these will be secreted by endocrine cells, meaning our glands that secrete hormones, which will help us through certain physiological processes such as growth, development, metabolism, and reproduction. An example of one of these hormones would be insulin, which of course would help regulate our blood sugar. Our building blocks of our proteins are called amino acids. So this is our monomer that is going to help build up into a protein. And we have 20 different arrangements utilizing these amino acids. We will always have a central carbon though, which we call the alpha carbon, an amino group attached, a carboxyl group attached, a hydrogen, and lastly a group that we call R, or the side chain, because this is our variable group that is going to differentiate each amino acid. So I mentioned we have 20 of them total, and there were 2 rare new amino acids that were discovered recently, and that is selenocysteine and pyrrolysine. So this slide is a little bit overwhelming because we've got all 20 of our amino acids here. 9 of them are going to be essential, meaning that our body cannot produce them. And it would include ones like isoleucine, leucine, and cysteine, but we won't go through and memorize all 9 of them. And they have a chemical nature of that side chain to determine what characteristics the amino acid will have. For instance, you can see that these amino acids have been placed into groups such as polar or nonpolar. Some are going to be acidic and others will be basic. Now, to refer to these amino acids, we can either use a single uppercase letter or a three-letter abbreviation such as V or Val for valine. And we could take these amino acids and link them together to create a chain. So again, amino acids are the building blocks of proteins. So when we are able to have them go through a dehydration synthesis reaction, we are going to create a peptide bond, is what we call the covalent bond between amino acids. And as we add more and more amino acids, we are going to call that chain a polypeptide, because poly means many and peptide is referring to our peptide bond. And the side that is going to have that nitrogen group is going to be called the N-terminal, whereas the carboxyl group will be called the carboxyl terminal. Next, let's move into protein structure. We've got 4 different levels that the protein has to go through in order to be functional. So first will be the primary structure, then secondary structure, tertiary structure, and lastly, quaternary structure. So let's discuss each one, starting with primary, of course, and this is just that unique sequence in the polypeptide chain. So we've got all these different amino acids that form peptide bonds with one another, and this one happens to be unique to insulin. Once we change any amino acid in here, it's going to change the entire protein. So how do we get these amino acids to all be correct in a row? It's encoded within our genes. So that essentially is going to create our protein, giving it a specific structure and function. Now, when we take a look at our hemoglobin, the example that your book gives over here, we see that there is a change in just one of those amino acids, and that change results in what we know as sickle cell anemia, which is an inherited disorder. And we've got 4 different hemoglobin molecules, so essentially 4 different proteins that have to come together in order to make hemoglobin. And so we replace our glutamic with valine, which forms long fibers that then distort the normal biconcave disc shape that our red blood cells have, and instead will create, I'm losing my mouse here, there we go, will create this crescent or sickle shape to the red blood cells, which then will clog our blood vessels, especially in the areas of our capillaries, our smallest blood vessels in our body, leading to things like breathlessness, dizziness, headaches, and abdominal pain. Now for the secondary structure. This is just local folding that's going to take place in our polypeptide. So we have 2 most common forms, which are the alpha helix pictured here and the beta pleated sheet structure. The shapes are held in place by hydrogen bonds between oxygen atoms and carbonyl groups. And so depending which amino acids are used in here will determine those bonds that take place to give either a beta pleated look or a alpha helix. Now for our tertiary structure, this is going to add that 3-dimensionality to it because of chemical interactions between the R groups of the amino acids. We'll see that certain R groups will repel each other and others are going to be attracted to each other, sometimes even forming ionic bonds. We'll see interactions between things like the cysteine side chains to form what's known as disulfide bonds or linkages, and sometimes have our hydrophobic R groups lie more on the interior of the protein because they're avoiding water or being around the surroundings. And then we also have our hydrophilic R groups lie more on the outside. So when a protein loses its 3D shape, it may not be functional. So this is a crucial step in creating the structure of the protein. Last structure is quaternary, and this is when we have several polypeptides or subunits that are going to come together. So for example, we created this beta globin polypeptide, and it's going to come together with other globins in order to create our hemoglobin molecule. And we tend to see that there are weak interactions between these subunits that helps to stabilize the overall structure.